The apparatus used in the BOD experiment includes a dissolved oxygen diometer, 6 BOD bottles, a diluted water container, a BOD incubator, and a measuring cylinder, while for the materials required are water samples, distilled water, and 4 types of chemical reagents, which are phosphate buffer, magnesium sulfate, calcium chloride, and ferric chloride solutions. To begin the experiment, all BOD bottles were first cleaned using distilled water to remove any contaminants. Each bottle was then filled with 50 milliliters of the water sample collected from the lake. The dilution water was prepared earlier by mixing 4 reagents into distilled water and aerating it. It was then added into the BOD bottles to top them up. Next, the sealed BOD bottles were placed inside a BOD incubator set at 20 degrees Celsius for 5 days. Before incubation, the initial DO of the sample and a blend was measured using a calibrated diometer and labeled as DO note. The value that we obtained for the DO note is 6.59 mg per liter. After 5 days, the final DO reading was recorded by using a calibrated diometer and labeled as DO5. The value that we obtained for DO5 is 5.93 mg per liter. Finally, BOD5 can be calculated by using the standard formula.
This is the apparatus of ammoniacal nitrogen test using Nestle method. Sample cells, serological pipette, beaker and mixing cylinder, spectrophotometer, ammonia nitrogen reagent set, distilled water, and 3 water samples which collected from a lake with 3 different points. Before starting the test, the apparatus was washed and rinsed with distilled water to remove any impurities. The mixing cylinder is filled with water sample at 25 ml. For blank sample, the mixing cylinder is filled with distilled water at 25 ml. Next, 3 drops of mineral stabilizer is added to each cylinder and shaken carefully to mix. Then, 3 drops of polyvinyl alcohol dispersing agent is added to each cylinder and shaken carefully to mix. One ml of Nestle reagent is pipetted into each cylinder and shaken carefully to mix. Timer is set for 1 minute to allow reaction period. 10 ml of each solution is poured into a square sample cell. The program number 380N ammonia nest is selected. When the timer expires, blank sample is wiped clean and placed into the cell holder, then press 0 to get the 0 value. Prepared sample is wiped clean and placed into the cell holder and read button is pressed to get the readings of ammonia content. Repeat the steps for another 2 water samples.
Apparatus for COD test is spectrophotometer, micropipette, and micropipette tip. And then materials is COD vials, low range, distilled water, and water sample.
Before starting the COD test, it's essential to avoid contamination. Here, a small beaker is filled with distilled water. Without touching the tip, the micropipette is used to draw and release the distilled water. Next, I prepare the sample for the COD test. First, I remove the cap from the COD vial and hold it at a 45-degree angle to avoid spills and ensure accurate pipetting. Using a clean pipette, I carefully add 2 ml of the water sample into the vial. Once the sample is added, I immediately close the vial tightly to prevent any evaporation or contamination. Then, holding the vial securely by the cap, I gently invert it several times to truly mix the contents with the reagent inside.
Then turn on the reactor and set the temperature to 150 degrees Celsius and let it preheat. Open the lid and place the prepared COD vials into the reactor block. Then close the lid. Start the timer and digest the vials for 2 hours at 150 degrees. After digestion, turn off the reactor, take out the vial into the rack and let it cool.
I begin by selecting program 430 COD low range. Then I clean the blank sample cell with a tissue to remove any water spots or fingerprints that might interfere with the reading. After that, I clean the prepared sample cell and insert it into the cell holder. I press the read button and the COD value for the sample appears on the screen in milligram per liter. Lastly, I read and record the result for further data analysis.
For turbidity test apparatus, firstly sample cells or bottles, beaker, turbidimeter, distilled water and also water samples, which consists of 3 water samples. Firstly, all of the apparatus is cleaned with distilled water. Water sample is added to the beaker. Then, the water sample is poured into the turbidity bottle until it reaches to the white marking label at the neck of the bottle. Then, the value of turbidity is taken by using turbidimeter. All of the steps will be repeated using other water samples. Please ensure that the bottle is closed tightly by using the bottle cap.
First of all, we need to clean our sample tube with a tissue until our sample tube is clean. Switch on the turbidimeter. Make sure the turbidimeter is set to auto range. Put the sample tube into the turbidimeter slot. Close the lid and tap the read button. Wait the reading until it's stable. Record the reading. Take out the sample tube from the turbidimeter and clean up the material and apparatus. pH test apparatus is sample cell and pH meter, while material is distilled water and water sample.
For the pH test, I start by pouring the water sample into a clean beaker. After that, I carefully fill the sample cell with the sample, making sure it's filled to more than half of the cell's height.
So, this is the procedure of using pH meter. First of all, the pH meter probe need to be cleaned by using distilled water. After that, attach the pH probe into the stand while setting the sample cell that filled by water sample. Switch on the pH meter. Make sure the probe is properly dipped into the water sample. Tap read button and wait until the pH value is stable, which is the log symbol appear. So this is the result.
For suspended solid test, the apparatus and material used are sample cells or bottles, beaker, spectrophotometer, distilled water, and lastly water samples, which consist of 3 different points at the lake. The water samples is taken from section 7 lake, which nearby UiTM Shah Alam. Measure the suspended solids, clean the apparatus first. The water sample will be poured into a small beaker to be then transferred to the bottle. 10 ml of water sample is added to the bottle. Then, blank sample of 10 ml, which is distilled water, is poured to the small beaker and added to the bottle. Reading is taken using spectrophotometer. Then, same steps is repeated by using other water samples too. Here is the sample in the bottle.
I begin by cleaning both the blank and the sample cells using a clean tissue. This removes any water spots or fingerprints that could interfere with the accuracy of the reading. Once both cells are clean, they are ready to be placed into the spectrophotometer for measurement. I first set the program to 630 suspended solids. Next, I insert the clean blank cell into the holder and press the 0 button to calibrate the device. After that, I place the clean sample cell into the holder and press the read button. I then record the reading for analysis.